introduce you, Laura. Here we go. Recording is on. Well, it's my pleasure to introduce Dr. Laura Charlanne. She's assistant professor at the University of Strasbourg. And Laura was first trained as a vet before completing her master's in evolutionary, functional, and behavioral ecology, where she studied the energetics of hibernation in the garden dormouse. And then she graduated her PhD in 2024 in August, and that was on the behavioral and physiological adaptations that allow southern elephant seals to perform extreme deep dives. And so her work focuses broadly on how animals cope with intense energetic and environmental constraints, which is perfect fit for the EcoMito community. And so that's why we've got her today. And she'll present her research who's been recently published in JB, which title is Ready to Dive, Early Constraints Help Juvenile Southern Elephant Seals Acclimatize to Aquatic Life. And so with that, Laura, up to you.
Yeah, thank you. Thank you very much. Hi, everyone, and thank you for having me for this journal club. As Jill said, I did my PhD on southern elephant seals and basically about everything, every constraint in their life and how they deal with this constraint with their physiology and behavior. But today I'm going to focus on the acclimatization to aquatic life and more precisely how we believe that the early constraint juvenile experience can help them to acclimatize to this aquatic life. So as Drew said in the presentation, I've always been interested in species that live in extreme environments, well, extreme, at least that face important environmental constraints and how they adapt to these constraints, and especially in environments where we, as humans, we will need a little help to feel comfortable with. So in that line, I found that southern elephant seals are very, very impressive and very interesting. So if you don't know them, elephant seals are one of the biggest fossils. Well, the one with the biggest sexual dimorphism. You have the northern species remaining in North America near the Californian coasts. And I've been working on southern elephant seals, so the polar species that can forage until Antarctica. And so this species, there's around 300,000 individuals. They are on the red list of the IUCN. And you have here the main colonies, including Macquarie, Marion Island, and Kerguelen Island, where there is a colony I've been working on. So why do I think they are so impressive? It's because they will regularly face, like all the life, important constraints. First, they will spend months at sea foraging. So they will perform deep dives until 1,000 meters in apnea and up to 1 hour or even 2 hours for the biggest males. And this continuously, so during this period and this period, while they are foraging. And so they are foraging in apnea, which means they will hunt, but despite the low oxygen availability, which is a big constraint. And then when they come back on land, they will come back twice a year, first for the reproduction, during which you have the female give birth, you have the lactation and fighting for the males, which is very energetically demanding. They fight and only the biggest and the strongest male will have access to the females. And then you have another online period, which is a molt, which is also extremely costly. It's a catastrophic molt, which means they will renew both the skin and the fur. And this in a polar environment, so they have a huge energetic disequilibrium at that period because they don't eat at all. They stay on land while doing these costly things. So it means that along all their life, they will face this disequilibrium. So during the foraging period at sea, this disequilibrium between oxygen availability while remaining active during the dives, and on land this disequilibrium with energy intake, low or no energy intake, and really costly activity. And so my entire PhD was about understanding how they can rebalance this disequilibrium along their life through their physiology and behavior. So this is for the adults, which is quite impressive. But for the juveniles, it's the same, and it's even a bit worse because they will be born on September and between September and October. You have 3 weeks of lactation, so the females will feed the juveniles, and then the female lives, and the juveniles remain on land for 3 to 5 weeks. It's the post-weaning period, during which they fast, but at the same time, they progressively increase their activity. They start to go to the sea, they start to perform small apneas. So they already face this important energetic disequilibrium because of this increase of activity while fasting. And then what's really impressive is that they will brutally transition to an aquatic life and immediately perform extreme dive. Well, it's a bit less extreme than the adults, but they will go to the sea for several months and start foraging and nearly as like the adults. So they will face also this challenge of low oxygen availability while foraging. And so we believe that this post-weaning period is very important, is critical for these juveniles because of this energetic disequilibrium they face and because you have this brutal transition. And so we think that maybe something happens during this period, maybe something about the maturation that could help them to transition from a terrestrial to an aquatic life. So that's why we were very interested in juveniles. So we talked about the challenges of foraging, of low oxygen availability, but it's not the only challenge these animals have to face when they dive. Here you have the representation of the dive. So during a foraging trip, they will do several repeated dives, and they will come back to the surface only a few minutes to breathe. And here you have another challenge, the oxidative stress challenge, because when they breathe at the surface, you have this ischemia reperfusion phenomenon, and this huge amount of oxygen comes to the organs that can lead to the production of reactive oxygen species and lead to oxidative stress if these species are not balanced by the antioxidant defenses. And if you have too much reactive oxygen species, that can lead to cell death, well, at least trigger deleterious effect on the cells. And so the question was, is there something during the post-weaning period that could help the juveniles to transition and to cope with the constraints of prolonged dives they will face during the next forage trip, including the oxidative stress and low oxygen availability? So in another word, how do they progressively become extreme divers and what are the physiological consequences? So to investigate this, we investigated juveniles during the post-weaning period. We sampled 5 females during the post-weaning and 6 adults, females also, during the reproduction, so during the lactation. And because we were interested in this balance between oxygen availability and foraging, we investigated the swimming muscles in this species. So to have a look at the experimental design, so we sampled both juveniles and adult females, and we did some muscular biopsy in the field, and we immediately performed a high-resolution respirometry to investigate the mitochondrial function of their muscles. And back in France, we also did gene expression and proteomics. At the same time, we took some blood sample that allowed us to investigate the oxidative stress part of the article. So all of this to try to understand better to better understand the cellular and molecular adaptation to hypoxia. So just a few words about the field work. So it took place in Kerguelen Island, a few days from La Réunion by boat, and more precisely in Port-au-Français. So here you have a photo of the site. I did the surgeries, so the sampling of the animals here, and the high resolution respirometry in this shelter. Not that far, so we didn't have to wait too long before running the analysis. Sorry. So we needed first to sedate the animals, then we weighted them, and we did the muscular biopsy, which was a bit challenging because we have almost 8 to 10 centimeters fat, so it's not that easy to sample. That's why we had this really small amount of muscle, even though for the females they are like 300 to 400 kilograms, but it was difficult to get more than 20 to 30 milligrams muscles per sample. Also because we wanted to do really a small wound because it's difficult to follow them after the surgery. So basically, I took 2 samples. Both were put on ice and on biopsy until I stitched the wound, and one part was frozen at -80 before later analysis in France, and so the other was used directly for the respirometry in the field. So in the shelter, I prepared the fibers, I did the permeabilization, and I put around 2 milligrams to 3 milligrams in the Ouroboros to perform the measurements at 37 degrees, the body temperature of the animals. And we ran 2 different protocols, so one using carbohydrates and the other using fatty acids. Just a quick reminder about why we are looking at the muscular cells and why we are looking at the mitochondria. It's because we were interested in the energy production and the oxygen consumption of their muscles. So we have to look at the mitochondria, which is the biggest producer of ATP, and especially through the respiratory chain. So just to have a quick reminder, ATP is produced at the end of the chain through the ATP synthase. And this needs an H+ gradient, which is maintained by the electron transport chain. So you need electrons, usually provided by the NADH, for example, which comes from the mitochondria itself, from the citrate cycle, through some substrates coming from the carbohydrates, or from fatty acid if you have an intermediate step, the beta oxidation. And we talked about reactive oxygen species, and we know that the mitochondria can produce some reactive oxygen species, especially through complex 1 and complex 2. So here we also wanted to take into account this balance between ATP production, oxygen consumption, and reactive oxygen species production. Through an indirect method, because in the field we were not able to estimate directly with the respirometry the reactive oxygen species production, unfortunately. So to remind the terms I will use in the results with the example of the carbohydrates. So we started by adding carbohydrates to the Ouroboros. So we produce NADH, we start to stimulate the electron transport chain, and we measured the leak first. Then we added ADP and we add the phosphorylative oxidation of the complex 1, so OXPHOS-C1 in the results. Then we stimulated the complex 2, and so we have OXPHOS-C1 plus C2. Then we inhibited the ATP synthase, so we have the maximum capacity of the mitochondria, C1 plus C2. We inhibited complex 1, so we have maximum C2. And then we inhibited everything, and we had the residual respiration. So to make sure that we all have the same terms about the results. And if we look at the results, so here you have the oxygen consumption in juveniles and in adults for the fatty acid and for the carbohydrates. And we didn't find any differences using these values, leak, OXPHOS, between the 2 ages, no age differences. But if we look at the respiratory ratio, we can calculate different ones. This one, so OXPHOS minus leak on OXPHOS, which is the OXPHOS coupling factor, it could be used as a proxy of mitochondrial efficiency. We found that for the fatty acids, it was higher in the juveniles than in the adults. And that was a bit surprising because in northern elephant seals, they found the opposite in juveniles and adults, but this was done in males. And this ratio was the same for the juveniles, but it was very high for the males. And so we think that it could more reflect the energy demanding period in adults at that moment, because the males, they remain very active during the reproduction, because we are in the muscles here. So they remain very active, while our female, they are lactating, so they don't move at all. And so it makes sense, as the juveniles here, we said that they progressively increase their physical activity. So yeah, we think that this difference in mitochondrial efficiency could more reflect the energy demanding period in the adults, rather than a direct effect of hypoxia. And so yeah, as we looked, we said, okay, if there's a difference in the mitochondrial efficiency, we wanted to look if there was something at the protein level and the gene expression. So that was, we did the proteomics and gene expression when we were back in France. And this allowed us to compare all the proteins with the proteomics. It was really strong to compare all the proteins that were different between the adults and the juveniles. We had 2,000 identified proteins and more than 200 that were with an IG effect. And among these 200 proteins, we found that some of the electron transport chains, so complex 1 and complex 4, were increased in juveniles, as well as many enzymes, many proteins involved in the citrate cycle, the beta oxidation, and substrate import. So that was consistent with this higher mitochondrial efficiency we found in our juveniles. We also found some evidence that we had a higher mitochondrial response in these juveniles. First, because we had an increase in this energetic cell sensor, and an increase in these two proteins that are related to the mitochondrial response, mitochondrial biogenesis and morphogenesis. And this is also might be consistent with an increase in their physical activity, because we showed in other juveniles, not this one unfortunately, but that they progressively increase their physical activity along the post-weaning. So that would be consistent with this. And so now the question is, if we have a difference in mitochondrial efficiency, is there something with the reactive oxygen species generation? So, which leads to the other part of the study, focusing on the blood this time, because reactive oxygen species can be balanced by the antioxidant system, but if not, it can impact different things in the cells, lipids, proteins, and DNA. And so in this study, we were able to quantify the consequences of reactive oxidative stress on lipid and on proteins. We looked at the plasma 8-isoprostaglandin and the protein carbonyls, and we couldn't find any difference between our juveniles and our adults. So no difference in the consequences of oxidative stress, which can mean two different things. Maybe they produce the same amount of ROS, or maybe one group is exposed to more reactive oxygen species but have higher defenses. And again, the proteomics and gene expression allowed us to look a bit more at this, because if we look at how reactive oxygen species are produced, so they can be produced by the mitochondria that can lead to oxidative damages, but this reactive oxygen species will stimulate antioxidant defenses through the NRF2 pathway. And these antioxidant defenses will prevent oxidative damages. And we found that these defenses were decreased in juveniles compared to adults, which means that the adults have higher defenses, and probably because they are more exposed to reactive oxygen species. And that can be a consequence of different things. First, of hypoxia, because adults regularly perform extreme dives, so they might be exposed to hypoxia more than the juveniles that only perform small apneas at that moment. And this was consistent with the hypoxia adaptive response, because we looked at all the pathways and the response to hypoxia with the proteomics, and we found that this adaptive pathway to hypoxia was decreased in juveniles, so higher in adults compared to juveniles, as well as the consequences, so the hypoxia response, including the metabolic consequences, or the effect on oxygen stores. So yeah, this is relevant with this consistent with the higher exposure to hypoxia by the adults. But we also found, like I said earlier, that this pathway, the energetic cell sensor and the mitochondrial response, was higher in juveniles. We said that it could be a consequence of physical activity, but it can also be a consequence of hypoxia. So maybe the small hypoxia the juveniles start to experience can contribute to this pathway. And some authors suggested that having a higher mitochondrial response could help the individuals to face, to cope with the constraint of hypoxia later in life. So having this response having this kind of response in juveniles could be a benefit, might be a benefit later in life facing hypoxia. And there is another challenge we haven't talked about yet. It's the caloric restriction, because the juvenile during that period, they fast. And we know that fasting can also increase reactive oxygen species generation and oxidative damages, so stimulate this pathway, and can also stimulate this response through the energetic cell sensor and the mitochondrial response. So we know we have this mitochondrial response in the juveniles, but we don't really know which challenge, or it's probably a combination of all the different challenges they experience at that moment that lead to this response, which could be an advantage to face hypoxia later in life, but not only hypoxia. Maybe it could also be an advantage to face other energetic constraints that they will regularly face later in life. So that's why I said again in this cycle, we saw this mitochondrial response during the post-weaning, and we believe that the early exposure to all these energetically demanding conditions may activate pathway and adaptation in these juveniles to prepare them to face or to cope with the constraints of prolonged dive later in life, but also to the other energetic challenges that we regularly face. So just a few words about the perspectives and the limits of this study. First, the sampling, of course, because as you saw, we sampled the females during the lactation. That was a big issue, but we were not able to sample them just after their return on land because they were pregnant. We didn't want to sedate them before they give birth, and not really early, not before the pups can be alone a little. So we are aware that this sampling reflects also the lactation and not only hypoxia, the effect of hypoxia. So that was the first thing. Also, we don't investigate the sex effect, but it would be very interesting because the males, they don't have the same energy, they don't have the same activity than the females at that moment, and they also perform deeper and longer dive. So it would be really interesting to have the comparison. Also, I would have loved to cross behavioral and physiological data on these juveniles. We had this accelerometry data on juveniles, but not this one, why it would have been really great to see how amount of time all juveniles spend in the water, remaining active. It would have been a beautiful story to have everything together, and will be great to do this in the future. And also, it was difficult to interpret this data with only one sampling during the post-weaning. At least it would be great to have one sample at the beginning of the post-weaning period, and then at the end to have really the change in the muscles of the juveniles, because one sample is quite difficult, especially because you don't really know the age of the animals. You can estimate them through the fur, but it's really difficult to be sure if they're in the middle of the post-weaning or really close to their departure to the sea. So yeah, that was challenging, and it would be great to have several sampling along the post-weaning. So that's it. Lots of people to thank on this project. Or first, thank you for your attention, and thank you for inviting me. And a big thanks to all the people that worked with me in the field, and either in the field and in the lab, because we did a lot of things, and I couldn't do this alone, of course. So thank you, everyone.
That was a great talk, Laura. Thank you very much.
Thank you.
Well explained. I'm really glad you actually explained a little bit of their life, at sea and on land, because I don't think it was obvious for everyone, certainly not me. Does anyone has any question for Laura? You can raise your hand or just unmute and ask any question. No, not yet.
Sure, I can start off.
Yeah, Neil.
Yeah, so Laura, that's it. No, that was an awesome talk. It's so great to see all of the like how much work you did for this project. There's just so many things sampled. I guess yeah, my question is on kind of it's like a two-parter and it may be way off, but roll with it anyways. So you show all of the regulation of the key pathways
Mhm.
and you show some things that are going up in the juveniles or at least are greater in the juveniles.
Mm.
I was just wondering, because of the way you measured muscle fiber typing, right? So you know the number, but not the size. Do you
Yeah.
think that maybe what you're seeing is that in the juveniles, they have smaller muscle fibers? So it looks like there's more of the, sort of mitochondrially related genes simply because they're taking up a greater proportion of the fiber
Mm.
area. And then as they become more mature, they have larger muscle fibers, they have more, muscle machinery thing is in there. So you're just seeing an artifact of this increasing muscle fiber size.
Yeah, that's a good question because we were not able to do some histological analysis, but we estimated the mitochondrial density, and it looked like it was the same for juveniles and adults. So at least we know that we have the same mitochondrial density, and we also have the same fibers type because we looked at the
Yeah.
fiber types, and it was both for both type 1, major type 1, and little type 2A. But
Okay.
I believe Karin, you raised your hand and wonder whether you had a question or.
I had a similar question that Anil and Laura, you replied to my question. But I had another question, so thank you, Laura. It was a great presentation and a great project, I think. So I have a bad internet connection, and you might have said it to start when you explain about the biology of your model. When they go diving for feeding, how long do they stay in the water? Because I feel when I look at your schematic, I feel like they stay 3 months. So I suppose sometime they go back, first they go up and down in the water, and then they go back on land every so often.
yeah, they go. So the dive usually they last between 20 minutes to 1 or 2 hours for the biggest, but they will stay at sea for several months. They won't come back on land. They come back only for the reproduction and for the molt. So they will just perform repeated and repeated and repeated dive during months.
Okay, and about the ischemia reperfusion, when the knowledge we've got come from human biology, most of it, where it can have impact on the brain function mostly,
Yeah, that's
which
a good.
is so maybe is it because some tissue are more sensitive to drop in oxygen or to zero oxygenation, or some mitochondria stop to consume oxygen at higher level of oxygen, so the level of oxygen in the cell doesn't decline that much? So my question is there do we know of variation differences between tissue in the sensitivity of the of the hypoxic condition, and maybe
Yeah.
the blood doesn't care actually?
Yeah, that's a good question. We know that most of the oxygen stores in this species remain in the muscles, very few in the blood, and some also in the brain. So we know that the brain and the muscles are a priority. I don't know if I can say this, and that the other are kind of shut down. However, there's no study that I know considering the sensibility of the tissues. I just know that something is going on the brain, and it's a colleague of mine that was on the field, Erwan. And yeah, there's something definitely about vascularization of the blend that is highly maintained, for instance, to maintain the oxygen in the brain, as well the same in the muscles. But yeah, for the other tissue, unfortunately, I don't know. But I know that yeah, the brain then is a priority also.
Perfume. I don't know if you had any other question, Karen, but oh no, you're all good. I might just Antoine. I'll actually give priority to Neil because he typed his message in the chat before you raised your hand. So Neil was just asking if you knew whether there were any difference between juveniles and adults in the density of mitochondria. If you actually had tested that, I think
No,
that's
yeah,
related to
we,
Neil's.
no, we didn't test it this precisely, but we looked at the amount of, if I remember, the amount of citrate synthase, and it was quite the same. So we used it as a proxy to say, okay, it might have the same mitochondrial density.
Awesome.
Mm.
Antoine, your turn.
Yes. Thanks, Laura, for your talk. So it's possible that I already asked you this question when you were maybe like practicing for the PhD defense or something, because the results you presented on the efficiency, so that's basically being calculated from leak and oxphos respiration. But since you were like in your protocol being able to dissect a bit between like complex 1 and complex 2, I was wondering if you looked at the contribution of complex 1 and complex 2, like because that can influence like the ATP, basically like what amount of ATP is like ending up.
Yes, yes, I remember we talked about this. I didn't present it here, but yeah, you can see that the complex 2 contribution, that's quite surprising, but the complex 2 contribution is higher than complex 1 in both cases, and either for juveniles and for both juveniles and adults. And yeah, that's surprising because usually you improve more the efficiency when you improve the complex 1. But there is recent study that suggests that complex 2 could contribute to the what they call the reserve respiratory capacity, which is the difference between the maximum respiratory capacity and the basal one. And so complex 2, if it's increased, could improve both the electron transport chain and the entrance of substrate in the cell citrate cycle. So both age could benefit from a higher complex 2, we think, because they are facing fasting, physical activity, well, energetic constraints, important energetic constraints. So yeah, but yeah, I agree, that's surprising.
Okay, thanks.
If no one has any questions, or Neil, maybe you unmuted
Oh
yet?
yeah, sorry.
Go for it.
I have just a for fun one. So this is more just to see if you encountered similar issues when using fatty acids with the mitochondria. How did you notice the difference between using palmitocarnitine and octanylcarnitine?
Hmm.
Did you find that one was more stable than the other
No,
in
I
terms
don't
of the
remember.
respiration rate?
I don't remember seeing something between the use of these two, because you do?
Yeah, I think for me, I noticed so I did exactly the same as you. I added palmitocarnitine first with malate, and then when I added octanylcarnitine, it just smoothed everything right out.
Okay.
And a couple other people I've spoken to have sort of noticed the same thing. So I'm just wondering if I don't know if it's like a different transport mechanism or what's going on, but it seems as soon as you add in octanylcarnitine, everything just is so clean. And I tried just like one run without ever adding it, and it's that variation stays kind of it persists throughout the whole run.
Okay. Well,
So it's just I
interesting.
was just yeah, no, I guess there's not really any like biological question. It's more just a technical thing if you noticed it as well.
Yeah, I'm gonna have a look at the curves again, and I will check. Okay.
Or if anybody else has encountered this and knows what the heck's going on, that'd be awesome to hear from someone.
Haven't really used oct, but I know that for palmitoyl, yeah, we do also have some sort of almost as if it was trying to reach an equilibrium, right?
Yeah, and it
Uh,
never does.
yeah, and it kind of never does. That's right. Um,
Yeah, we only use here exclusively oct. I think maybe because of that issue. I don't think we have any like thoughts or reasons as to why what's happening with Palmitoyl, but yeah, the oct is just much smoother, as you're saying.
Hey, so Laura, so you mentioned that the muscle was mainly type 1, right? Oh, mainly type 2. Yeah, okay, yeah, no, that makes sense. I actually thought that the dorsal was mainly kind of like type 2 muscle. Is there any other, I guess, justification as why you chose that muscle? Like I know it's important for diving, but it's also for reachability, the fact that you're maybe even safety way where, instead of punching through like a pectoral
No, yeah.
muscle where you can actually get hit or something, like is it also part of the
Mm.
uh
Yeah, it's very easy to access, and also because it's on the back, well, when you sedate them, to put an intravenous needle on the back, so you need them to remain in their belly. So
mhm.
it was really easy to access at that place. Mm.
And did you think or could you, I mean, it's limiting when you're doing studies in the field, right? There's only so much you can do. But did you also think about exploring it to other tissues like adipocytes, for example? Would you expect kind of the same trend between juveniles and adults in other tissues, or like what's your thoughts on that?
Yeah, that would be interesting to do this on the adipocytes, but they don't have if they don't have the same amount of fat at that moment, because the females, when we sampled them, well, during this period, they were lactating, so really in the middle of the lactation, they were really low amount of fat, well, lower than the juveniles. So probably the fat reserves were not the same between both. Yeah.
Okay. And then if no one had, oh yeah, Jamie had one. No.
Ah,
Oh no, Karine. Yeah, there you go.
right. I was simultaneous with Antoine, sorry.
Yeah.
Um, yeah, you had this, you emphasize your limitation, which is rather a perspective of a low sample size. And is it something you can expect to do one day to sample 40 juveniles and 40 adults and to analyze their mitochondria, or just never gonna happen?
No.
It's a perspective or it's just a limitation and we stop there?
Maybe if you do this, maybe like 10 animals per campaign each year, maybe you can have more individuals, but not at the same time, not the same year. I don't think so, because
It's
it's really
ethical issue or a biological access constraint or just human resources or what's the issue?
everything. First, ethical aspect,
And
because
money.
it's difficult to have more than five, seven individuals. Also the timing, because it will take one day to do just one individual, because you can only do the surgery early in the morning when there is no wind, or it's very complicated. And so yeah, the females, they will remain only 3 weeks, and we don't want to sample them too early during the lactation, so maybe we have only access to them during 10 days, 10 to 15 days. So it's not that much. And also for the juveniles, if we want to sample them at the same time also, they won't last. Well, we have to just have to do this at the same time for all the juveniles if we want to have them to have something like at the same time, if they are at the same physiological state. And yeah, so it's difficult, I think, to do more than I think between 10, maybe 10 more during one campaign.
Okay. And my question is because, let's say, the good diver, they have good mitochondria, if that's the prediction. Do you think it's possible to relate? I suppose some individual dive longer or deeper than other, or some are faster swimmer. Is possible to relate their swimming performance or diving performance to their mitochondrial property? I mean, for that, you will need a good sample size, but also I feel like you've got the data on the diving. And is it something you can project to do?
Yeah, it would. If I have the data on the diving, it would be interesting to relate. And yeah, you can look at different parameters in the mitochondria. Well, actually, Neil did a study on the ducks, and for instance, the citrate synthase is a good proxy of the swimming performances. So yeah, I think we could do that if you have both the swimming parameters and you have access to the muscles, you can sample the same animals.
Mm. Thank you.
Antoine, you can.
Yeah. And considering that they are dying at the moment because of avian influenza, I think it's unlikely you can sample more. But no, I had a bit like a funny question that Laura, you will know from a PhD defense of last week. If you had an elephant seals in front of you being able to talk, what would you ask to the elephant seal? Like what secret?
I will ask Juvenile what the strategy do they how do they increase their time at sea? You know, this balance between the body condition and the time they can spend at sea, because they have limited amount of fat, and if they have more fat, they will spend more time at sea. They will have most time on land before going to the sea. While if they are in bad body conditions, they will be forced to leave early. So I will ask, is there a balance between the time, the energy you spare and you spend training, or do you wait? Is it the time that it is important, or the amount of time you spend in the water? Is it the training that is critical, or is it the time?
Yeah. It was a fun question last week at the PhD defense on king penguin, and I really liked it because we work on different kind of animals, and there are a lot of we don't know. There are stuff that we can study, there are stuff that we can't, at least at the moment, and that gives some nice perspectives, I think.
That was a good question.
It's based on the assumption that the king penguin and the seals, they know better about their physiology than we know about ourselves as a scientist. So they are very smart.
They know their limits as well. Actually, has anyone looked at ROS production during dive in general? Like, do you know if they, I mean, you know, certain mammal actually accumulate succinate during like ischemia, in hypoxia. Do we know if it also happens in seals? Like, has anyone looked at this?
Not during dive, but there's a group at St. Andrews that I've been loosely chatting with. So I don't know if anything will come of it, some of our like discussions, but they actually have harbor seals that they're able to work on.
Mhm.
And so they can take samples, blood samples, and I think they're allowed to take 2 biopsies. So you could do maybe like pre-dive, post-dive for kind of muscle biopsies, but you could take blood throughout. So we've talked about doing something like that, but I honestly have no idea if anything will come of it.
Okay,
But
yeah.
they basically train them to come into this giant swimming pool, and they teach them to repeatedly dive, sort of bribe them with fish.
Okay, because that was a question, or I thought it would be really interesting to see what's happening during the dive and while they're actually anoxic, but how would you get the samples? It's like, how does that even work? And so what is the mechanism of them getting the blood samples?
Complex, but they have these sort of attachments that they can put on that takes like automatically takes blood
Mm.
every programmed amount. So again, I've only seen it in theory. I haven't got to see it happen in practice, so it'd be difficult. And also, how I feel like in order to capture the sort of the raw state, you'd have to get those samples and freeze them immediately before major changes start to take place. So yeah, there's some logistical challenges, but at least it's potentially could happen there.
Mm.
Who knows?
Maybe I missed. Wouldn't this make sense then that of regulating complex 2 before in the juvenile phase to avoid the succinate-driven reverse electron transfer during hypoxia reoxygenation, which you got more efficient complex 2 and all the TTA cycle enzymes you showed? Wouldn't that work actually as a strategy to avoid ROS production?
Yeah. We couldn't hear you really well, Enrique, but I believe what you said was actually, yeah, they've got more complex too, so they would avoid succinate accumulation by chewing up more succinate.
Yeah, I
Yeah.
don't know. But they also have more complex 1. So the reverse electron transfer at complex 1, I would say there's potential for more damage there, but there's also more flux through complex 2.
Mm.
So would that make sense as a priming mechanism to avoid reverse electron transfer? I don't know.
Could be. We saw the other way around in sharks, in anoxia-tolerant sharks, in
Yeah.
fact, so they would suppress succinate
Yeah.
oxidation.
Mm.
But one or the other mechanism could also be either, it could be beneficial either way, I guess.
Neil, I'm in a basement in London. I don't know why I sound like that. I'm far from the deep oceans.
All right. Well, if has anyone any other question? Because if not, we might actually wrap up the session. So thank you again, Laura, for accepting the invitation. It was a great paper, a great talk. We're really pleased to have you on board. And thanks everyone for tuning in as well, and asking plenty of questions. It was really interesting, so we wanted to know even more. And we'll be uploading the recording on YouTube really soon.